Hello everyone. Today we are going to talk about disaccharides and polysaccharides. This lecture follows the lecture on chapter 11.1 where we talked about monosaccharides. So when we talked about monosaccharides, we discussed how the monosaccharide, here glucose is shown, in its linear or its Fischer conformation could undergo a hemiacetal reaction to form a cyclic structure. And here the aldehyde and the hydroxyl group on carbon number 5 participate. And depending on which direction the nucleophilic attack occurs, when the ring closes and this carbon number 1 that was the aldehyde now becomes a new anomeric carbon. When the ring closes, the OH that's formed as a result of that reaction could end up below the plane of the ring, in which case we call it alpha, or it could occur above the plane of the ring, in which case we call it beta. And notice that these are equilibrium arrows. So if you have glucose in solution, at any moment in time you have all three of these structures. So here's another picture here. So here this shows in highlighted in pink the aldehyde and the alcohol that participate in the hemiacetal reaction. And here the artist has taken this linear structure and then drawn it sort of bent around so we can see that that hydroxyl group on carbon number 5 has access to the carbonyl on carbon number 1. And again, we have two different isomers, anomers, that can form depending on whether the OH ends up below the plane of the ring or above the plane of the ring. So what this means is that we say if we had the alpha, we could go back to the straight chain form and then we could form the beta. So you can interconvert easily between these anomers. So that phenomenon is called mutarotation. So for example, if I started out with some solid alpha-D-glucose and dissolved it in water, then I'd have an aqueous solution of alpha-D-glucose. But if I wait a while, I will find that when the system reaches equilibrium, I've got 64% of beta-D-glucose and 36% alpha-D-glucose, and less than 1% is in the straight chain form, although this is a dynamic equilibrium. So we can tell that there must be some increased stability for the beta-D-glucose. So let's look at that. So remember, we've been drawing these monosaccharides as flat hexagons, but you know from organic chemistry that they have a chair conformation. They're not flat hexagons. It's just easier to draw it that way. So if I look at my Haworth projection right here, which is most likely how you're going to see it, and it is easier to draw that way, the sugar really, what it really looks like is it's in a chair conformation. The chair conformation, as you might remember from organic chemistry, is more stable than the boat conformation. The other thing that we can do is if you sort of tug on this end right here and then pull on this end right here, you can interconvert between two forms of the chair conformation. And again, you might remember from organic chemistry that when you have the most bulky substituents in the equatorial positions, that's the more stable form. So that means that the form on the left will be more stable than the one on the right. So if I compare alpha-D-glucose and beta-D-glucose, and if I draw them in their chair conformations, which is easy to do on ChemDraw, because this is a part of they draw the ring for you. If we look at the OH on the anomeric carbon, this is carbon number 1. For alpha, we see the OH is below the plane of the ring, so that means that this is axial. Whereas for beta-D-glucose, the OH is above the plane of the ring, so this OH is equatorial. So that's one of the reasons that beta-D-glucose is more stable than the alpha-D-glucose. The other reason is that this configuration right here allows the OH to be more highly hydrated in the beta form. So this is the case for glucose, but I don't want you to think that all sugars, all monosaccharides have a more stable beta form than an alpha form. It depends on the sugar. For other sugars other than glucose, there are some electronic effects that I won't get into here that might favor the alpha anomer. But for glucose, the beta-D anomer is more stable. So now let's talk about disaccharides. So a glycosidic bond is a bond that forms between two monosaccharides. It's a condensation reaction, and we can either get a cyclic acetal or a cyclic ketal depending on the monosaccharides that we use to make them, whether it's their aldoses or ketoses. So let's see how this works. I'm going to use 3 examples: maltose, lactose, and sucrose. So let's look at maltose first. So maltose is a disaccharide. It consists of 2 D-glucoses. So I've drawn 2 D-glucose. I've drawn alpha D-glucose on the left and beta D-glucose on the right. And what I'm going to do is I'm going to form a glycosidic bond between the 2 of them. So I'm going to take off this OH and that H, and that will give me water, and that's what I get. So that's my structure of maltose. So maltose is a common name. It is a condensation reaction, and we refer to this bond right here as an alpha glycosidic bond. If we wanted to name this precisely, it has a very long name, so we have O right here. That refers to this oxygen right here. And this is the sugar on the left. It's an alpha D glucopyranose. So remember from the last lecture that pyran means that the glucose is in its 6-membered ring form. And then there's a 1 here because the bond is between carbon number 1 of this glucose, the glucose on the left. And then I draw an arrow, and there's a 4 because that's the carbon number 4 on the glucose on the right, and the glucose on the right is D-glucopyranose. So maltose is the common name, but this is the name that we would use if we wanted to specify exactly the configurations of the glucoses and how they are linked together. Okay, so let's look at lactose. You might be familiar with lactose. You find it in dairy products. It is a disaccharide, and it consists of glucose and galactose. So I've drawn beta D-galactose over here. So there's the anomeric carbon. So it's beta, so the anomeric carbon, the OH, is above the plane of the ring. And then I drew alpha D-glucose over here on the right. I'm going to draw it again in this configuration because this is where I'm going to form the glycosidic bond, and I want them to be next to each other. So again, if I take off this hydrogen right here and this OH right here, then I can form a glycosidic bond that looks like that. So this is a beta glycosidic bond, and that's because the anomeric carbon on the galactose over here on the left, the OH, before I made the glycosidic bond, the OH was above the plane of the ring. So again, lactose is a common name, but if I wanted to name it precisely, I'd say O, so that's this oxygen right here, beta, because the galactose, the carbon number 1 was in the beta configuration. So O, beta D, galactopyranose, and again, that means that the galactose is in its 6-membered ring form 1, there's carbon number 1, and then an arrow is 4, so it links to carbon number 4 of the glucopyranose. So that's the structure of lactose. Sucrose is table sugar, so you might be familiar with sucrose. It consists of alpha D-glucose and beta D-fructose. So I'm going to redraw that. So I've drawn them on top of each other. That's not really exactly how sucrose looks, but it's just easy for me to draw it this way because I want to show you how I'm going to form the glycosidic bond. I'm going to take off a hydrogen here and an OH here to do my condensation reaction. And there is my sucrose right here. So I copied this from Wikipedia a number of years ago because I wanted to show you what sucrose really looks like. So I, again, didn't realize when I copied the structure I'd be copying the animation, but there it is. And you can see a little bit better, I think, what the structure of sucrose really looks like. It doesn't really look like this. I have all the bonds are formed properly, but this doesn't really give you a sense of what sucrose really looks like. But I do want to emphasize that we are forming a glycosidic bond between the anomeric carbon of glucose. So here, if we look at its name, O, alpha D, glucopyranose. So this is the glucose in its 6-membered ring form. There's the OH. It was alpha below the plane of the ring. And then there's a 1 arrow 2. So there's carbon number 1. This is carbon number 2. And this is beta D fructopyranose. So the OH in the fructose, the OH was above the plane of the ring. And we say furan because fructose is in its 5-membered ring form. So now we're in a position to understand a term that we call reducing sugars. This is a redox reaction. We can take any compound that has an aldehyde and we can oxidize it to a carboxylic acid in the presence of copper, which becomes reduced. So this is a test for reducing sugar. So there's a solution called Benedict solution. So it consists of copper sulfate in a basic solution. So the copper sulfate's a blue solution and the oxidation state is +2. But in the presence of an aldehyde that becomes oxidized, the copper becomes reduced to the +1 form, and the copper 2 oxide is a solid. It's a red solid. So if you take your glucose and a Benedict solution and heat it up in a heating block, you will form the red copper 2 oxide. So reducing sugars, in order for this to be a positive test, you need a free aldehyde. And in the redox reaction, the sugar is oxidized and a metal ion is reduced. So we call these reducing sugars because they are able to reduce the metal. There's another test called the Tollens test, and it's similar. You use silver. Here, again, the aldehyde is oxidized to the carboxylic acid. And the silver, you have an ammonia solution of silver. Here you have silver in the +1 oxidation form and actual silver plates onto the glassware. So you get a silver metal. So the silver becomes reduced. So again, you'll get a positive Tollens test if you have a free aldehyde group that can be oxidized, and then that is able to reduce something else. So that reduces the silver from +1 to 0. So if this were a normal classroom, I would ask you a top hat question, and I would ask you, is glucose a reducing sugar? So think about that for a second and pause the presentation. So the answer is yes, it is. Sometimes I had trouble convincing students of that in the past. They would look at this and they would look for a free aldehyde and not see one. But then I would remind the students, and I'll remind you here, that remember, if I present to you a structure of alpha-D-glucopyranose like we show over here, that ring can open up. And if the ring can open up, there's your free aldehyde. And then the free aldehyde is able to react with the copper sulfate or the silver, whatever test you happen to be using. So that will oxidize this aldehyde group to a carboxylic acid, so that shifts the equilibrium until pretty soon all of your glucose is oxidized. So the next question is, let's move to the disaccharides. Is maltose a reducing sugar? So think about that and pause the presentation and I'll come back. So here again, you're looking for an aldehyde. You don't see one in this structure right here, but because this is carbon number 1, that's the anomeric carbon, you can actually open this up and get an aldehyde right here where I've indicated with the red arrow. You can't over here, because here the anomeric carbon is involved in this glycosidic bond. So this carbon number 1, that anomeric carbon is tied up. But this one can open up, it's not tied up. So maltose is a reducing sugar. Let's ask the question again for lactose. Is lactose a reducing sugar? So think about it and then I'll come back. Well, the answer is yes, lactose is a reducing sugar. And again, this carbon number 1, this anomeric carbon, is tied up, so that can't react, but this one can. This one is not tied up in a glycosidic bond, so you could open up the ring right here and get your free aldehyde, and then you'd get a positive test. So lactose is a reducing sugar. How about sucrose? Is sucrose a reducing sugar? So take a minute to think about that. So the answer is no. Sucrose is not a reducing sugar, and the reason is that you have two sugars, and both of them, the linkage is with both of their anomeric carbons. So you've got the anomeric carbon here, carbon number 1, the anomeric carbon right here, and since both of them are linked in a glycosidic bond, they're not available to have the ring open up. And one of the things you can do is, if you noticed when I presented the names for you, that this didn't say fructofuranose, it said fructofuranocide, and that's name is on purpose. So the if you see the name of a sugar and it ends in side, that means no aldehyde or no reducing end. So sometimes you can tell in the name whether it's reducing or not. So how do you tell? A summary. How to tell if a sugar is reducing. So this is a test that I did. So this is from my lab. And I did this with my natural science class, and I had glucose, fructose, maltose, and lactose. And all of them were positive, as I just showed you. And that was sucrose. So sucrose stayed blue. So that was a negative test. So for disaccharides, look at the structure. If you have one anomeric carbon that's not involved in a glycosidic bond, the ring can open up at that point and the disaccharide is reducing. And that's because the cyclic form of the sugar is equilibrium with the straight chain form as described by Mutarotation. For monosaccharides, you'd expect aldoses to be reducing because they have an aldehyde. You'd expect ketoses to not be, but I did tell you that one of these tubes right here is fructose, and you do get a positive test. So the reason for that is that if you add fructose to a Benedict solution, what happens is the ketose actually gets isomerized to an aldose, so you still get a positive test. So even though you learned in organic chemistry that you can't oxidize a ketone group, and you're right, it will still give you a positive Benedict solution test because the ketose gets isomerized to the aldose. So that's the long answer. The short answer is that all monosaccharides are reducing. So let's now talk about polysaccharides. They are polymers of monosaccharides. We can have heteropolysaccharides or homopolysaccharides. So heteropolysaccharides consist of 2 or more types of monosaccharide, whereas the homopolysaccharides just consist of just one. And that's going to be our focus in this class. We want to talk about the 2 types of homopolysaccharides. They're structural and storage. So let me give you some examples of these. So let's talk about cellulose first. For cellulose, the monosaccharide is beta-D-glucose. So you have somewhere between 300 and 15,000 glucoses, and they're all beta-D-glucoses. And the bond is a beta-1,4. So I drew 2 betas next to each other right here, but they're obviously not going to be able to form a bond very easily. So what I did is I redrew them. So here's one beta-D-glucose over here, and then I flipped this one so it's upside down. So now I'm able to form the glycosidic bond where I pull off this OH and that H, and now I have a glycosidic bond. So I have a beta glycosidic bond, and it's a beta-1,4. So this OH was in the beta configuration, and this is carbon number 4 on the upside down glucose next to it. So that's called a beta-1,4 glycosidic bond. So when you do this, when you get your hundreds or thousands of beta-D-glucoses together, this is what cellulose looks like, and it forms these sheet structures. And here you can see the glucose in the chair conformation. So here we are in the right side up, and here we're in the sort of the upside down configuration. And you have a big network of hydrogen bonds that keeps them together. So here's another example of a structural homopolysaccharide. This is called chitin, and the monosaccharide here is N-acetyl D-glucosamine. So if we look at this, you can recognize glucose, but instead of the OH right here at carbon number 2, you have an NH2, so it becomes glucosamine, and then there's the acetyl group right here. And those are also beta-1,4. So here the beta, the OH on carbon number 1 would be above the plane of the ring. And again, if you wanted the carbon number 4 to react with it, you'd have to flip it upside down. So you have this alternating sort of right side up, upside down. And the places you would find chitin would be in the exoskeletons of animals like lobsters. But I learned kind of recently that mushrooms also have chitin. So So Cook's Illustrated lets us know that mushrooms are very forgiving. You can quickly saute them for a few minutes and they're delicious if you like mushrooms, which I do. Or you could roast them for an hour and they don't fall apart. So the reason for that is that they have chitin, so it's a very heat stable kind of thing. So that's why you're able to cook mushrooms in lots of different ways. Okay, those are structural homopolysaccharides. We want to focus in this class on storage homopolysaccharides. So if you are a plant, you will have two types of homopolysaccharides, amylose and amylopectin, and those are the plants that we like to eat, like all this bread and pasta that's shown in this picture. In our bodies, we store, our storage homopolysaccharide is glycogen. So amylose, what does it look like? Its monomer unit is alpha D glucose. So all of the monomer units are alpha D glucose. It's not like cellulose, which were beta D glucose. These are alpha D glucoses. So anywhere between 100 and 1,000. And here we have the alpha 1,4 glycosidic bonds. So here I've drawn two alpha D glucoses, and again I'm going to pull off an OH here and an H here, and I get my my amylose. So here, this is drawn as a bracket with an N right here. So this just shows us that we have a whole string of alpha 1,4 glycosidic bonds. So that's what amylose looks like. Since you just have this linear polymer, though, amylose is interesting in that it can kind of fold up to form this helical structure, and what happens is that, for reasons I don't completely understand, an iodine could fit in here. And when it does, the iodine turns blue. So this is a starch test. I don't know if you did this when you were kids. You might remember in general chemistry, we did an experiment called the iodine clock, where you added a starch indicator and you added a compound that produced iodine in a reaction, and it would turn blue all at once. So that was the basis of this. So here, this picture shows apples, which have cellulose. So you drop an iodine on there, and the iodine is yellow, so you don't see a positive test. But here, if you take a slice of potato, potato has amylose in it, and the iodine orange drop turns blue. So it's interesting, though, it's actually not I2, it's an I3 minus ion. So what happens is in solution, I2 and I minus react to form I3 minus, and you get an I3 minus ion, and that's what forms the blue color. Okay, so that's one form of starch that we find in plants. That's amylose. A second form is amylopectin, and this has branches. So, and it's bigger. It's up to 1 million glucoses. And again, all alpha D glucose, and it has the same alpha 1,4 glycosidic bonds. But you also have these branches. This is called an alpha 1,6, where this is an alpha, an OH here, and this would have been the CH2OH up here, and this forms an alpha 1,6 linkage right here. So if this were a normal classroom, I would ask you to identify the reducing ends, so end or ends. So think about that for a second and see if you can identify them. Well, it turns out when you have a branch structure like this, you only have one reducing end. So these are non-reducing ends, again, because there's their carbon number 1s, and they're involved in the glycosidic bonds. But if you look over here, and this is involved in a glycosidic bond too, but this one is the only one that has a ring that could open up. So what that means for the structure of glucose is if I expand this and draw this highly branched structure, you only have one reducing end that's shown here in green. In blue, all of these other ends are non-reducing ends. The reason we're interested in that is that in a few chapters from now, we will talk about glycogen metabolism, and some interesting chemistry happens at the non-reducing end. So we'll just tuck that away in our brains for later on. So here's an up-close structure of the alpha 1,6 branch point. So again, you have the OH, which is an alpha glucose. The OH is below the plane of the ring, so it's alpha, and you form a glycosidic bond with a CH2OH, a carbon number 6, and that gets you the branch points. So that was starch. It's a mixture of amylose and amylopectin in plants. In animals, we have glycogen. So you might have noticed I even used the same picture. So glycogen and amylopectin are very, very similar. Um, the monosaccharide unit is alpha-D-glucose. It has the two types of bonds, the alpha-1,4 and the alpha-1,6. The differences are the number of glucoses here is about maybe 50,000 glucoses, and the branch points occur more frequently than amylopectin. But you can see why I was able to use the same picture to illustrate both things. So glycogen in animals is stored in two places, in the muscle and in the liver. And there is our reducing end. Okay, so let's talk a little nutrition now. So if we're going to eat these starchy things like potatoes down here, our diet consists of starch if we eat things like potatoes or wheat or things like that. We don't really have a lot of glycogen in our diet. I told you that glycogen is found in liver and muscle, so you shouldn't eat liver. Liver's bad for you. Don't eat liver. But if you ate a steak, for example, that's muscle. But we don't. What happens is slightly after butchering, the glycogen degrades. So we don't really have much glycogen in our diet. Most of the starch that we have in our diet is plant-based. So there are enzymes called amylases, and they will hydrolyze all of those alpha-1,4 and alpha-1,6 glycosidic bonds so that we can get alpha-D-glucose. And this happens, it starts happening in the mouth actually, and then is completed in the small intestine, and then the alpha-D-glucoses are absorbed. And when you carry out digestion, it has to be a monosaccharide. Even a disaccharide is too big for our small intestines to absorb. Well, what about cellulose? So cellulose, remember, has those beta-1,4 bonds. So the amylases won't work on those. So if you eat a cabbage, which consists of cellulose, you would need a cellulase enzyme to digest it to its beta-D-glucoses. So single-stomached organisms like ourselves, though, don't have cellulases. So if we eat anything with cellulase in it, like that cabbage, we can't digest cellulose. But if you think about cows are ruminants. They have a digestive organ called a rumen. And this is a quote from Michael Pollan. He describes it as a digestive tank in which a resident population of bacteria dines on grass. So cows eat grass, at least they're meant to do it that way. Our feedlots often will feed cattle corn, beef tallow, protein supplements, and antibiotics, which does not sound appetizing. But you've heard maybe a lot about grass-fed beef. So cows are meant to eat grass, and their rumen digests the grass very well. So the cows themselves don't have the cellulases, but the bacteria that live in the rumen do. So when the cow eats the grass, the bacteria provide the cellulase enzymes that are able to digest those beta-1,4 glycosidic bonds. So we are not ruminants. We don't have a rumen. We don't have those bacteria. So we can't digest cellulose. So they don't provide energy for us, but they do serve as fiber. So they aren't digested. And because they're not digested, they add bulk to the stool, which speeds its passage through the gastrointestinal tract. And there are some studies that indicate that diets high in fiber are protective against some forms of cancer. So when a nutritionist says fiber, that's just basically an undigestible carbohydrate. So just to finish with a few more interesting concepts. So there are proteins called glycoproteins where sugars are attached to proteins. And we have two different types. We can call them O-linked or N-linked. So if you remember, serine is CH2OH. We can do another condensation reaction. So this is N-acetylglucosamine, and we can attach a sugar here. So that's an O-linked sugar. Or this is an N-linked sugar, so this is asparagine, and this would have been an NH2, and again, another good place to do a condensation reaction. So these sugars are on the outside of the cell, and they're useful in protein sorting and recognition, which you might cover in one of your biology classes. We cover it in biochemistry too. It's beyond the scope of this course. But glycoproteins might serve a protective function for cells. You might remember elastase. Elastase was one of the enzymes we used in our sequencing, and we saw that we could digest proteins with elastase after small amino acids like glycine and alanine because of that small pocket that it had. But elastase is actually a glycoprotein. So if we look at the structure of elastase, these are the structures of the sugars you see on the surface of that enzyme. The last thing I'm going to leave you is, I'm sure you're familiar with ABO blood types. So these are defined by different oligosaccharides. So if we look at our little key right here, we have galactose, N-acetylglucosamine, N-acetylgalactosamine, and fucose, and different arrangements of the sugars on the surfaces of the cells define the blood type. So we have blood type O and B and type A, and you can see that they have different combinations of sugars on the surface of the cells. So if this were a normal day, I would then pass out an IFAT quiz for you to take. I have posted the quiz on Sakai, so you should take that quiz and make sure you can solidify your understanding of these concepts. And later on, I will post the answers and answer any questions for you in office hours. So I hope you enjoy this and talk to you soon.